Okay, let's start. You're all witness that it's 5 past. So I will, now that Sergio is here especially, I will try to limit the questions from the front row, except from Eric, because I'm following the example of Laplace. So so now, okay, so here I will talk about work that well, I will talk mostly about classical work that we haven't done, but if I get to it, I will talk a little bit about some work that we did with a student, Simone Blanco Malerba, and a postdoc, Felix Franke. Okay, so I thought that I'm probably the only person here who's also working on some neural questions, some question of neural coding, looking at neural data. And since this workshop, this summer school is about cognitive imprecision, after all, cognition is implemented in the brain. And so I thought it would be not a bad idea to have one lecture about the culprit, the brain. And but this lecture will be very uncomfortable, I think, for you. One, it will be uncomfortable by necessity because I will cover a lot of things in a very superficial way without going into the details. So this lecture could be called something like 12 monologues with no hidden message at all. And the other thing, it will be uncomfortable by design because I will go into the realm of neurons that is much more messy than I think the realm most of you are accustomed to. There's a poem by Baudelaire where he describes how wonderfully the albatross is flying, but then when the albatross comes on the ship and is walking and you see it from up close, it's very awkward and uncomfortable. So going down to the level of neuron is a bit like this. So, so until now I talked in the previous lecture, various other speakers have talked about the idea of maximizing some performance or minimizing some loss while limiting some cost that is incurred when doing this. And this optimization is done over this quantity, which is a density or a probability distribution over internal representations conditional on physical observations, on stimuli. Okay? Now, as I pointed out in the previous lectures, and especially in this lecture, I will want to think of these representations as implemented by brain activity on which we base our actions. And already when you say this, you have to go beyond the kind of simple models we've considered because your actions will change both the stimulus and the representation. So for example, I'm moving my eyes, and this will, you're actually looking at me now about 3 or 4 times a second your eye is saccading without you noticing. And so you're modifying the stimulus by your own action. Now the internal representation is also changing in non-trivial way as a result of action. So your visual cortex, for example, will fire differently if you're walking than if you're sitting. Okay? Now, none of this I will talk about, but I just want to point out that this simple kind of feed-forward process is very restrictive. So often we will think of an action giving rise to a reward, and also the reward might depend not only on the stimulus, but on latent causes of the stimulus that in the models that we saw this morning, for example, were described as some prior over the stimulus. And so in this talk, and as I mentioned this morning, and we will illustrate this internal representation should be thought of as a random representation, not as a deterministic representation. So if I show you several times this owl from Picasso, the firing of your neurons will be somewhat different each time you see this image. Okay? So what we'll be concerned in this lecture is try to understand a little bit how this representation depends on the rewards and depends on the prior. And one proposal I will want to illustrate in this respect is what some people call efficient coding proposal, which is that the neural representation optimizes some performance subject to some biological limitations. So this is very similar to what we saw before, The difference is before we had an information cost that Ariel didn't like, and now we'll replace this by some actual biological cost. Okay? So what's just in a cartoon, what does that mean? So imagine that you have a signal, this continuous trace over time entering your visual system, for example, and you see that most of the signal stays within that range. Occasionally you have a deflection lower. Well, if you have your internal representation has some allowed range that it can occupy because your spikes cannot fire more than something, and I think Mike will discuss some models of this flavor in his lecture on Sunday, then you might want to, and also this representation is subject to noise, then you might want to devote a large chunk of your representation space to describe the region of the stimulus where action is going on, and a small chunk of your allowed space to describe where there's not much action from the stimulus. And so you will distort, as compared to S, you will distort your internal representation in this way. So that's, in a cartoon, the idea of efficient coding. Okay. So now the outline, which is probably much too optimistic, I will try to give you some facts about neural signaling and about the cost of neural signaling. I'll then illustrate some implication for neural coding, emphasizing the fact that neural activity is noisy and asking what efficiency can give rise to. Then I will want to just illustrate very quickly that these approaches are very simple, leave aside a number of important points. And if I have time, which I probably won't, but if I have time, I'll want to discuss also decoding. Everything that I'll discuss here, like what I said this morning, will assume that you decode optimally. Okay? So information is encoded in neurons in some way, but we decode it abstractly according to some optimal scheme. In fact, the decoding is also done by neurons. You have several layers of neural processing, and so decoding also can be imperfect. And finally, which I doubt I'll ever get there, I want to connect to the morning lecture to show you how neural noise can affect memory. So to connect back to the lecture this morning. Okay, so let's start with neural signaling. So this is a pyramidal neuron, about in cortex, in a cat actually. So about 80% of your neurons in the cortex are of this kind. This is an electrode that is measuring the activity of this neuron, and the hand that holds this electrode is the hand of David Hubel, who was a great scientist, wonderful person also. I'll come back to him. And you have a lot of neurons in a cubic millimeter. In a cubic millimeter of your cortex, so these are processes of neurons, axons and dendrites. In a cubic millimeter of your cortex, you have about 4 kilometers of axons and dendrites in each cube millimeter of your cortex. So how do neurons communicate with each other? The way they communicate is if one neuron is excited, there will be a potential difference that will travel along the axon that will reach the synapse, and at the synapse will excite the postsynaptic, the neuron it's connected to. I'll come back to this with some more details, but this is what it looks like. This neuron sends this axon contacts in the synapse with this postsynaptic neuron, and information is transmitted between the two. And you can measure through this electrode these spikes that are generated by neurons, these potential deflections that you see here in a recording from David Hubel. So I should think of a neuron as sending these discrete messages to another neurons. I can measure these discrete messages. Every time I see a spike, I can do a tick. And then I can show twice the same stimulus, and I'll have a bunch of responses to the two trials where I've shown the same stimulus, and there will be some differences between the two because there is some noise. And so I can describe the activity of this neuron as a number of spikes per unit time, and that will characterize the response of this neuron to some stimulus. Okay?
Question? Yeah. That's measured at the synapse?
No, this is measured.
Or the other?
You see how it's measured. It's like this. So the electrode is, you bring an electrode close to the cell and you measure the potential difference.
So the issue is that the other neuron may be due to some other changes, whereas the synapse you can know where it comes from, no?
Okay, I will give you a class on electrophysiology in private.
So
Yes.
it's just the frequency that changes, but the size of the spike is the same?
That's right,
Okay.
with some small notes. But just, there are many ways to record. You can record, it's very difficult to record at synapses, but you can record with ways that to some extent are informative of where signals are coming from. Okay, and we can discuss this one. But this is just a general recording of the whole potential difference undergoing in the soma of the cell or at the axon hillock. Okay. So what happens at the synapse here? What happens is that you have the action potential, the spike coming through the axon. This will trigger mechanisms that will release these vesicles that are filled with neurotransmitters like glutamate. They will be liberated in the liquid, in the synaptic cleft between the two cells. They bind to some channels. These molecular channels open and ions flow in and out of the postsynaptic cell, creating a new potential difference. And if you have many of these arriving, then this cell will be excited and in turn produce a spike that it will send to its postsynaptic cells. Okay? And if you want to see what it looks like, this is what it looks like. And don't be fooled, I worked a little bit at some point on synapses. These postsynaptic domains at the synapse are made of hundreds of different molecular species, are ruled by very complicated dynamics and have 1 million parameters. So it's a very powerful computational machine that in this talk we'll completely ignore. But I think it's good to have a sense of how complex and rich things can be. So to emphasize this, we will mostly ask questions at the level of neurons or networks of neurons, but the nervous system spans tens order of magnitudes in size, and at each level you have very complicated systems. At each level, in fact, you have networks of many interacting agents, either molecules, kind of synaptic domains, neurons, different brain areas. So at each level you have very complicated networks of interacting agents.
What is CNS?
Central nervous system. So now, how much does it cost you to use your brain? So you can measure the glucose uptake. That's the main fuel of the brain. You can measure the glucose uptake. About 90% of what is taken up by the brain is used. So your brain uses about 15 watts. So your brain is like a dim, very dim bulb. Now the interesting thing is that this still represents about 20% of your resting metabolic energy, okay? Whereas your brain is about 2% of your body mass. So it's a very expensive organ. In fact, people have compared the brain usage of energy, which is here. It's somewhat higher than the heart and much higher than all the other organs. And in fact, 1 gram of brain, listening to me, it consumes about as much energy as 1 gram of muscles if you're running a marathon. Okay? Now, even more impressively, in the blowfly, only its photoreceptors, so the first cells of the entry point of the visual system, use nearly 10% of its total energy, measured in oxygen consumption. And that's not all the consumption of the eye, because the eye in a fly is so heavy that it uses an appreciable amount of its muscular energy to just carry its eyes. Okay? So there are many ways in which having a brain is quite expensive. So where does this cost come from? Well, a primary reason why it's costly is the following, is that when you have a spike traveling, what happens is that successively channels will open along the axons, and ions will flow in and out to create this potential difference that you can measure, but that will disrupt the equilibrium that existed. And you need to draw back the ions out that came in and put the ones that went out inside. And you do that with chemical pumps that use ATP, and the ATP is generated by glucose. So 1 molecule of glucose will give you about 30 molecules of ATP. And the same goes at the synapses. After these ions enter, if you leave it like this synapse after a while will be filled. The potential difference will go to the resting potential. You won't be able to signal anymore, so you constantly have to re-equilibrate. Okay? So this is the energy accounting for rodents, which was estimated. And it's estimated that about half of the energy is consumed by action potentials, by spikes. And about a third of the energy is consumed by pumps working in the synapse. And then a small amount of energy is used to keep the cells in its resting state. Okay? So what we see from here is that spikes are very costly, because of course these also results from spikes received at the synapse. So this is estimate for rodent. For humans, there's a very recent estimate from 2021, which looks a bit different. So as I said, you use about 15 watts in your brain. Most of it is just for heating. And about 4 watts is used by the cortex. And a large amount of that, about 3 watts, is for signaling, so mostly for spikes. And if you look at this diagram, this whole amount, so 37% of the energy, is used in your white matter. And white matter means axons that send information from one area in the brain to a remote area in the brain. And the reason it's consumed so much energy is you want to make these axons fast. Okay? So the fact that you're using so much energy to do something fast is maybe a hint already that energy enough is not alone a sufficiently detailed constraint. Maybe time of computation should also be a constraint to consider. I'm not going to consider it, but I'm just making this remark. Now I'm sad to break it. Yes?
You don't have time to stop.
Sorry?
You don't have time to stop.
I'm sad to soon I will not have energy either. I'm sad to break it to you that you're not the fastest neurons. There's faster than you. There's a shrimp. It is faster than humans, and the reason they have such a fast neuron is that it controls the pump, and they can act as pump very fast to expel water and evade from predators. So that's an example where time is quite important. Now there is this amount of energy, which they call sin mod, I don't know why, but this is basically for maintenance and growth of cells, and I think it's important and but neglected in most of the kind of accounting of energy that is used more computationally. So how do neurons now report information? So this is a recording from Hubel and Wiesel, and they pioneered this way of thinking about the brain, which is you show a stimulus and then correlate the neural response to the stimulus that you've shown. And they measured in cat initially, then monkey, the activity at the back of the cortex, which is here, which is the primary visual cortex. So that's the second station after the eye. Your retina responds to light. The axons in the retina, which is the optic nerve, sends messages to the thalamus, and then the neurons in the thalamus sends their axons to the visual cortex. And what they found is that these neurons here are responds most vigorously to a bar of a given orientation. So if you show to this neuron that is being recorded here a horizontal bar, it doesn't respond. But if you show this oblique bar, it responds a lot. Okay? And so you can do this experiment for many orientation of the bar. And what you find is what I will call throughout the talk, because I'm accustomed to a tuning curve. And this tuning curve is telling you what's the average spiking response to a given stimulus. So here we're organizing the stimulus as along one parameter, which is the orientation of this bar. And as you vary the orientation, the average response of your cell in your visual cortex varies like this bell-shaped curve. Okay? Now, this is the mean response, but there's a lot of variability around this response. So this is the responses of neurons to a movie, I think, and this is, I think, 150 or 200 repeats of the same movie. And you can see some temporal organization. You see these vertical lines, but you can also see that it's extremely noisy. So if I blow up this segment, this is 1/10 of a second. This is the time, about the time it takes you to recognize a face. Actually, you can do it a bit faster than that. So for the brain, it can be viewed as a fairly long time, yet you see very little structure in the firing. So these are firing in response to the same stimulus. Okay? So they look very random. And in fact, you can look at the variance, you can pick a window of time, count the number of spikes on average, and look at the variance. And what you see is that the variance grows a bit faster than linear. So it's a bit more random than a Poisson process. Okay? Now, this is a different way to illustrate this curve. This is just a polar plot of this curve. So this neuron is responding most vigorously in this when the bar is moving in this direction, not when it's moving in this direction. So this is the meaning of this polar plot, it's just this. But now I want to illustrate for you the variability, again, of these orientation cells in visual cortex in response to these moving bars. And what you see here is an histogram of the spike count. So this is the spike count. And you can see that this histogram, you can see that the histogram has some width, okay? And so the spike count will vary quite a bit from trial to trial. And again, you can look at the variance of the spike count as a function of the mean, and you find something that is a bit higher than Poisson. You can explain it with some model, but that's not important. Okay? Any questions? Okay, so why are neurons variable? I'm not going to discuss it quantitatively, but I want to make some remarks of what this randomness, where this randomness is coming from. Well, the first thing is that there's biophysical noise. So as you said, spikes are roughly of the same size, but they result from opening and closing of channels, which fluctuate, under thermal fluctuations. So sometimes you have a bit greater number of ions that come in, come out. So you have some fluctuation of the size of the spikes. You have fluctuations of the vesicle release. Everything, all these molecular processes are fluctuating because of thermal fluctuations. Okay, so there's some intrinsic biophysical noise. The other reason is that any neuron is getting many inputs, as you were kind of pointing to, and in fact, in your visual cortex, you're also getting auditory signals. But those auditory signals will not help you to read this, but they will be represented in your visual cortex. So a lot of information is represented in any given population of neurons. And so when you correlate the activity of this population to one feature of the stimulus, the rest of the activity, what is not explained by the correlation, will look as noise. Yes?
If you suppose you show the Picasso owl audit every time, will the variance go down or will the variance stay constant?
Depends on the cell. You can see both.
So there is so some learn and others don't.
And some learn in different ways. The answer to, can you see, is it like this or like that? For any questions about biology, it's both. That's the universal answer.
Okay.
So the other thing is that you do not only have random inputs, you also have random modulations. So you have a neuromodulator that changes the state in which your brain operates. And finally, even if you didn't have any of these things, neurons are large dimensional coupled systems with nonlinearities. And so there is some evidence to believe that neurons, at least in some regimes, behave as a chaotic system, so that any slight change in the initial conditions will lead you to completely different trajectories of the activity. So I put it in gray because it's not some kind of fundamental randomness, but for all practical purposes, it is randomness. Okay?
But something has to settle off the chaotic dynamics. Is it one of the other three things, or
well, so you have in your cortex about 10 billion cells, all of them have some non-zero firing. So it could just be a self-sustaining system. You see, you're constantly. You see what I mean?
So if a neuron misfires or something, and then that sets
Yeah.
off.
Exactly. So, for example, just since we're talking about this, you have chaotic dynamics, and when you get general anesthesia, some of the chaotic dynamics goes into very regular oscillations. And so one thought about anesthesia is that you remove a lot of modes that are useful for conveying information by collapsing all these modes into one kind of oscillatory mode, and you kind of lose all the information. Okay, so now how does such a system will represent information that is relevant for behavior? So let's go back to this image, and let's imagine that I'm showing you these bars, and what I can do is to record the response of a neuron in your visual cortex. Now this is the mean response. What I'll record on any one trial will be one of these circles, and that's actual measurement. So for example, the one time I show this, there will be this response. Another time I show that, there will be this response. Okay? And so on the basis of that, you have to guess what orientation the bar was moving in. Of course, you don't have one neuron, you have a whole population of neurons. So if you present this stimulus, all of these will respond. Here I put the green dot on the mean response, but these green dots will be jittered up and down on every trial. Okay? And so based on that, you have to...
Each red curve is a different neuron.
That's right, exactly. Yeah, thank you. So this one way to quantify how well you can produce an action is to say, well, suppose that I want my action to just be to estimate the stimulus. So just a simple tracking problem, okay? So I will look at a pattern of activity in my brain. From this pattern, I will estimate the direction of the stimulus. I can then compare this estimate with the true direction of the stimulus, derive from this an error, and this error will be a figure of merit of how good my neural code is. Okay? So let's do this in a kind of somewhat hand-wavy way. So this is one neuron responding to this one-dimensional stimulus. This neuron's amplitude A has width W. Well, the noise that I will have on my estimate has this size, which is just the projection of the size of my noise through the slope of my tuning curve. Okay? So the variance of my estimation will be of order sigma divided by m prime, by this derivative, squared. If I have Poisson noise, this size will be of the order of square root of a, okay? And this derivative will be of order a over w, okay? And so my variance will be of order w squared over a, okay, for one neuron. Now I have many neurons, so suppose that I have m neurons, so the variance in my estimate will simply be the variance of the mean error, okay? And the mean error, so the variance of this quantity will be 1 over m squared times the sum of the variances from each neuron if I assume that neurons are not correlated. Okay? And so finally I'll get this. So if I have a population of neurons with these parameters, this is the error that I will make on estimating my one-dimensional stimulus.
So is this the same assumption that they are not correlated?
I will talk a bit about this later. It's not. They are typically correlated. Now, by how much and what's the structure of these correlations is the object of a lot of work. I've worked quite a bit on that, and typically they're not. But there are also cases where it's a legitimate assumption.
Sorry.
Yeah.
So the vertical axis is the intensity of the firing.
That's right.
And
Exactly.
Is it possible that instead of mixing the actual signal S with its neighborhood, or mixing it with somewhere on the left, always the same?
That's right. So you're absolutely right. So if I had one neuron, I would have this degeneracy. It's broken as soon as I have many neurons. So if I have many neurons, I could not. Imagine I have 2 neurons, then I lift this degeneracy.
Okay.
But there are more complicated coding schemes where you do have such inertia. I will discuss it later if I get to it. So now what we haven't specified is what is this M. And this M is simply, suppose that I now discretize, just for the sake of simplicity, my stimulus space, and I say on each spot of the stimulus I allow to have n neurons, and my neurons have a width wj. So I allow now my density of neurons to change along my stimulus axis, and the width to change. Well, in a given stimulus will be monitored by a number of neurons that is of the order n times w. Okay? And so if I replace this m here by n times w, I will get this nice result that the variance of my estimate will scale like w over n times a. Okay? Now, there are many ways to do this much more formally and much more cleanly and rigorously, and I will follow the Rubinstein doctrine and skip that. So I skip the more formal things, and because they give exactly the same result. But if you're interested, let me know, and in the break I can show you various ways to do that. Okay, so what does that mean? So it looks like what I would have to do to have a good estimate is either let my width to go to 0, or let my amplitude to go to infinity, or let my number of neurons to go to infinity, all of which don't seem to be acceptable solutions for the brain. Because we don't have infinitely precise neurons, we don't have infinitely many neurons, and our neurons don't fire infinitely high. Okay? But that's what this solution is suggesting. So what do these authors propose? They propose to add some cost. And the cost that they added was very much inspired by what I said before, which is simply the number of spikes, okay, with a prefactor mu. In fact, they also have to add a cost on the number of neurons, and you might legitimately ask why, because if only spikes are costly, you don't care if they're produced by 1 or 5 neurons, it will be the same cost. But in their approach, they need that term, okay? So what they want is to minimize some coding precision while also minimizing the cost spent in spike to encode with this coding precision. So you can see that it's very similar to the kind of rational inattention stuff I was talking about in the morning, only this is not anymore information-based cost, but it's a metabolic cost.
And where is this coding then? What part of the brain is performing this minimization?
So again, okay. So no. So I think there are two different. There are two possible ways to interpret what you're saying. One question is, does the brain kind of every time that it has to do a computation does some minimization like this? And that's certainly not what I'm advocating. What I'm advocating is that the brain has evolved in such a way that in the computation that it has to do, the system is set up in such a way as to minimize some quantity like this. Of course, this is a very simplified and idealized quantity. It will be, in reality, much more sophisticated than that. Okay, so I'm not claiming that the brain is doing an optimization computation. What I want to see is how should the brain be set up so that this be minimized. So how should the neurons, what should the properties of the neural population be such that you can code with limited metabolic costs, so that over your...
What you've shown us so far is how the neurons respond to the stimulus.
That's right.
You haven't shown us how the response to the neurons gets translated into an action.
Okay, so then there are three questions. So, because there's also an additional question to what you're doing is like, what gives rise to these responses? What gives rise to the response is not only the property of this neuron, but it's the property of the entire hierarchy of neurons that came before, okay, which is something I've ignored, and I will come back to it. Now, at least in the first part of the talk, I will assume that once you have this encoding, you can decode it optimally. Okay? This is an assumption that is done in most of these efficient coding models, the motivation being the following, that you have some peripheral systems like the retina, the primary visual cortex. Their job is to extract as much information as possible from the stimuli because they don't know what this information will be used for. Okay? Downstream from the retina or the visual cortex, you have an entire brain which has infinite computational ability and will be able to decode that information optimally for whatever action, say, the motor system wants to produce. So we're assuming here that the decoding is not a problem. Okay, and we're focusing on the encoding. I will return to this to show that there are cases where this assumption is experimentally not valid. Okay, okay, so this average coding imprecision, I have many choices. I could take the average estimation error, or variance. What these authors chose to pick, because it will give them solutions that correspond to, that match the data, is the average of the log of the estimation error. I could take any monotonic function. And in fact, you can show, if you have a lot of neurons, you can show that this is equal to the mutual information between the stimulus and the response, plus some terms that don't depend on the character of the response. Okay? So minimizing this average log of the estimation error is a proxy for maximizing the mutual information. Okay? So now, the additional step that they take to make it tractable is they look at the continuum limit. So I will make my discretization of my stimulus very, very fine. And now I can talk about the density per unit stimulus space of neurons. The amplitude of the response and the width of the response will all depend on where in the stimulus space my neuron is located, the preferred stimulus of the neuron. Okay? So now I can write this Lagrangian like this. I'm just translating these three terms in terms of our variables. But if you just minimize this, you find, again, a nonsensical answer of the kind I was discussing before, that you want this to become infinite. Okay? So these authors had to add one crucial assumption, which one can criticize, and I will criticize it, which is that they assume that the number of neurons used to encode a stimulus is constant. So locally, you're devoting the same amount of neurons to monitor a stimulus. That's the number of neurons that monitor a specific stimulus, and they assume that this is constant throughout the stimulus space. Okay? Now, if you do make this assumption, you find some...
neurons is the number of neurons involved in that. We're not looking at the total number of neurons.
No, total
so
number.
Why is it different than the lambda? Isn't that coming from the constraint that the number of neurons is less or equal something?
No, okay, so no, sure. But this is so this is a global constraint on the total number of neurons. This is a local constraint. So this is saying that the local density of neuron, the number of neurons per unit stimulus space, so in the case of the bar per degree, is the same. So any stimulus is activating a subset of neurons that are the neurons responsive to stimuli close to that stimulus. This volume of this subset, the number of neurons is fixed.
In S.
In S, exactly. Okay, so if you assume this local constraint, you get a very clean result. Namely, you get that the density of neurons should be proportional to your prior over the stimulus. Okay, this is the frequency with which a given stimulus occurs. The width of the neuron is inversely proportional to the prior, and the amplitude is constant. And furthermore, we already had calculated the estimation error or variance as a function of these parameters, and the estimation error is it gives you 1 over the prior if you combine these properly. Okay? Now, how could we check for this estimation error in an experiment? Well, what you can do is to ask people to discriminate two neighboring stimuli, and if these two neighboring stimuli are close enough that they're within this estimation error, they won't be able to discriminate them. So the discrimination threshold should be of order this estimation error. Okay, so that's one way to behaviorally get at this estimation error. And to come back to what you're saying, Eric, this is indeed assuming that you can decode it optimally.
Question?
Yes?
I lost track. Where does the prior come from?
Okay, so the prior is, I should have emphasized this. It's good you asked me. So everything here, how much spikes you use, for example, will depend. Let's say you use 5 spikes for stimulus A and 6 spikes for stimulus B. If you see stimulus D, 100 times, you'll use 600 spikes. And if you use stimulus A 5 times, you'll use 25 spikes. Okay? So the prior will be a weighting factor here. And the prior will also be here because you want to have an average of your uncertainty, not a uniform average. You want to weigh this average by the prior because, for example, if you never see a stimulus and this is 0, you don't care about coding it.
It's a bigger frequency of the various stimuli.
Of the various stimuli, exactly.
Now I'm confused because
Yeah.
frequency in the experiment. but
Frequency in
this
the
is- but
experiment.
This is done by evolution. This was done a million years ago.
Or in nature. I will show you an example in nature.
I mean, I was, probably a very stupid question. I was wondering, is the unit of analysis, I mean, here we are talking about neurons, but should it be the synapse or the neurons? I mean,
No, it's not. It's a very good question. So here we're assuming that these are the parameters that we can modify in this optimization calculation. And you're absolutely right that in nature, parameters much more refined than this and many more parameters can be dynamically changed or can adapt. And if you were God, you would write down the problem with all these parameters. So this is a brutal simplification that we're doing. I will show you some examples of how things can change over time, if I get to it. Okay, so as Tomasz or somebody was saying, so indeed these should be adapted over long time scales, and what we want to do is to compare these results in the context of naturally occurring priors over stimuli. And so what these people have done is they have an algorithm that tells them how many vertical and horizontal edges you have. These are picking up vertical edges, the red dots. So you can see that in nature there are a lot of vertical edges. And typically you have more horizontal and vertical edges. Like in this room there are many horizontal edges, many vertical edges. And so your images will typically contain many more bars that are vertical or horizontal than oblique. Okay? This is particularly strong indoors. It's also strong outdoors in cities where you have buildings. It's less strong in completely natural environment, but you still have some fluctuation of the prior. Okay? So people took this, they fitted it with a curve, so that's their distribution of the orientation of bars in nature. And then they went and recorded in the monkey these quantities, the width of the tuning curve and the density of the tuning curve. And here's the density of tuning curve of which the preferred orientation is S. Okay, so you can label neurons by their preferred orientation. This is the density of neurons with that preferred orientation.
So I guess experimentally, first of all, they realized that WS times NS is more or less constant, independent of S. So that thing was verified empirically or something. But otherwise, you won't be able to predict.
Well, I talked with the fellow who wrote the paper, and he really introduced it just for mathematical convenience, I think.
Okay.
There's the data.
So that's the data, which here you have no data. So this is just the theoretical curve. The white is no data.
But there are other stimulus domains where you do measure.
I will show. And then for the density of neurons, this is somewhat acceptable. So this is for monkey, but for Ariel, this is human. So this is the threshold, discrimination threshold for humans in terms of orientation. And here it looks like it adheres quite well with the theoretical prediction. And as Mike was saying, you can do this exercise for many different quantities because there are many cells that have these typical bell-shaped tuning curves with preferred directions. And for example, another example which I might come back to is an example in the auditory domain. So you can measure the naturally occurring acoustic frequencies. This is the distribution, and you can compare these quantities with the recorded data, which agrees quite well. So, so what are the implications for behavior, for perception that we can extract from such a way of thinking? Well, one thing that we could extract is this discrimination threshold, which I'll just show you some data about. But I would like to go a bit beyond this, and to go a bit beyond this, I will try to show you a somewhat more abstract picture. So let's start with, yeah?
So, in a sense, what you've shown, that whatever way you measure how many resources I'm using, is proportional to the frequency of what they need to respond to. So things that are more frequent, I'm using, that are twice more frequent, I'm using twice as many resources.
Yeah.
And you can measure it by amplitude, you can measure it by the numbers, but they all do the same, more or less. Is it a good interpretation?
Okay,
First level.
So it depends a bit, because what you don't listen to Mike. Okay. What you have to what
I get a lot of data trying to.
Well, you have a number of parameters, and you have a prescription of what you want to minimize, and then you'll get some solution in terms of these parameters. And roughly speaking, this solution is telling you how should I allocate my resources locally as a function of the prior, so as to spend as few resources as I can to have the result I want, which is what you were saying, right? But in the details that are in...
I was not looking at that. I was looking at the actual graph. I don't know the optimization problem. Okay. I'm just looking at the data, yes, and and all these graphs. That's more or less what they say, right? I'm comparing this graph to that graph. It's more or less the same. What the inverse?
I don't know what you mean. What do you mean? What they say?
Number of neurons is proportional to how often.
To how often they appear.
Right. But again, I never said that the resources is the number of neurons. We said that what is costly is the number of spikes.
I don't know, but each one of those graphs is a different measure of the resources used, and they all look the same, more or less. Okay, first order.
I mean,
okay.
I think the statement is too vague to have to be informative. So,
could be proportional to the square of that, for example. But that's not what you're saying. Yes, it doesn't increase like area. It increases like linear. Yes, for example.
I will show you.
Twice as many, twice as often, twice as frequent stimulus. I'm using twice as many resources, not 4 times.
And that's because I have this specific prescription, and the stimulus is one-dimensional, and I posed the specific constraint. It would not be the case in other conditions, and I will show you another example. So I don't think that this kind of rough way of reasoning is valid. So imagine that I have some population of neurons like this, and I follow this rule. So I imagine that they're organized in a way where the density is proportional to the prior. So here I'm imagining a prior that decays with S. So the density is lower here than here. Similarly, the width is inversely proportional, and so the width becomes bigger there, okay? Now, I'm free to stretch this axis locally as I want, and let me stretch it locally by a factor pi of s, okay? If I do this in this new effective stimulus dimension theta, sorry? It's not only, but it's not only that the prior is uniform, that's not the point. The point is that with these rules, now the population becomes uniform, okay? And so no matter how I'll encode with these neurons, I can think of this encoding as a one-dimensional abstract representation where my encoding variable will be rho, will be this effective stimulus dimension, plus some noise that now will not depend on theta, okay? Now this is a very kind of pictorial example I gave you, you can do it in general. In general, if I have a population of neurons and I want to extract a one-dimensional stimulus, I can find a manifold, a one-dimensional manifold in this high-dimensional space that will be most correlated with my stimulus, and that will become my effective internal stimulus dimension, if you want. Okay? But this is an example, a simple example of that case. Okay? You can also have another representation, which will be the inverse now of rho. It's theta inverse of rho, and in this representation, I would have the stimulus, the physical stimulus, but then I would have a noise whose variance depends on pi. Okay? And I just want to point out that this is important because it tells you how your internal measure of some stimulus relates to your noise. And this is a relevant point. This is related to, I think, in the talk of Mike or in the talk of Tomasz, where the Weber law was mentioned, and it was an outcome of how the internal mean representation relates to how the noise depends on the stimulus. This is another example of that. So the relation between your natural internal kind of dimension and the size of the noise, or the dependence of the noise, will have bearing on how you encode information. So now suppose that I have encoded now my stimulus in this abstract representation, which is noisy, and I'm extracting the value, an estimate of the stimulus from this representation. Okay? So this estimate will be noisy. We've already discussed this because rho is noisy. But will it, on average, give me a correct estimate? Okay? So, for example, to decide what my estimate is, I could have as a criterion that I want to minimize the mean squared error. And sorry, here I use a physicist notation. This means expectation, and I'm taking the expectation over S and rho, sorry. Well, then I know that the estimate will just be the mean of the posterior. Okay, so that's what I meant by optimal decoding. So the question is now, will this, on average, this coincide with S or not? Yes? So rho, so I said that I start with this population of neurons, and I said I'm free to represent it on any transform of monotonic transform of S I want. I decided to represent it in terms of theta. And in terms of theta, my population became uniform. So no matter what I'm encoding, I can always represent my internal variable as some rho, which is theta of S plus a noise that doesn't depend. So now does this S hat on average coincide with this S or not? So that's very easy to do. We just take this and we average over rho, and we can write this average as a weighted average over S, where the weight depends on the prior and on this function, which itself depends on my internal representation. Okay? So the picture is something like this. I will have to average S over a distribution that is the product of this function, f of S and S prime, and the prior. And what you can see is that if I do this product and then average S, because my prior has a negative slope, the mean will be moved to the left. Okay? And so the average of S hat will be to the left, actually, of S, will not coincide with S. Now, in this case...
What is S prime? Sorry, S prime denotes what?
Okay, so S, you're making an average over rho. So you're making an average over rho. So okay, S prime is here. So rho is conditional on S, on the stimulus that you've seen. And
So
S hat is conditional on rho.
So S prime is what would have been, okay.
Yeah. So, okay, so now in this case, what I showed you here is that my encoding, because of this rule, becomes worse in larger S's. Okay? So now, if you think about what that will imply for this function f, it will imply that it will not have this symmetric form. So this function f is essentially the overlap between my two representations, a representation of S and a representation of S prime. If I move to the right, my representation of S prime is more noisy than if I move to the left. More noisy means that this P will be broader. Because this P will be broader, this overlap will be bigger. Okay? And so this function will have a bigger wing to the right. Okay? And so now, if I now calculate the average, this average quantity weighted by the product of the prior and this function f, whether my bias, my average S hat, will be to the left of S or to the right will depend on which of these two forces is stronger, whether the prior pulls it more to the left or this difference in the precision of encoding pulls it more to the right. Okay? Now, this I did in a very pictorial way. You can do all this much more formally and actually obtain an explicit equation for the bias in terms of pi and the derivative of how much the encoding worsens around S. Okay? And if you apply that to this case, to this specific case, then what you will find, to this specific case, then what you will find is the bias will that the force of this f function will win and the bias will be to the right. Okay? So as a result, not only this efficient coding is giving you a result on what your perception uncertainty will be, it also gives you a prediction on the direction and the size of your average bias in your perception. Okay? And that can be tested experimentally.
So that is that the bias is towards less frequent signals?
Exactly.
Okay.
I have a question, maybe I'll start, but since everything in the end is correlated with the prior, what was the original reason to introduce both N and W and not, for instance, something like the product of these two? Like, I mean, like, in the end, to get your result, you are showing that it's really important that you can get this mono-dimensional, like this trick of like uniformization in a sense.
No, I said that, no. What I said is that you can always do this uniformization. You can always find a direction, a one-dimensional direction in your high-dimensional population activity that will be the direction most correlated with the stimulus. What I was saying is that in this case, with this specific prescription, I can make this simple transformation to give you what that direction is. Okay?
In general, it would be like a
Something else.
component analysis.
Exactly.
I find this surprising because I would have thought no matter what the encoding is, the decoding is optimal, and by law of the correct expectations, the average bias should be zero. The average of the posteriors is the prior, so I must be misunderstanding something here. I don't know what I'm misunderstanding.
But there is bias.
But where
It's
is
saying that it's zero on average.
Yeah. So it cannot. The bias cannot be everywhere in the same direction. So you'll
But
have.
you were taking average over S.
No. No. That's bias at a point S.
Oh, okay.
So So you're absolutely right that on average the bias would be zero. So I cannot have all the bias move to the right. So I'll have part of it to the right, part of it to the left.
So
Okay.
I misunderstood the
Yeah.
physics notation. Sorry.
No, sorry, conditional. Yeah, conditional. Yeah. Okay, so this is the experiment. So, they show you a bunch of these small oriented bars, and you're supposed to arrange these two white dots to say where the average orientation is. Okay. And again, the question is whether it's to the right or to the left of S. So, for example, if you have, so I'm here, let's say if I'm near, if I'm near the vertical, Okay, so this is the vertical orientation. If I'm on this slope near the vertical, does my bias go away from the vertical or towards the vertical? And what you see here is that it goes away from the vertical. So that coincides with the prediction of this model, that the bias takes you to the least expected result, not the most expected one. Okay? Now another comment which can be tested is, imagine this green curve where infinitely peaked around would have a zero width, then you would have no bias. And by the same token, if I have a bigger width, I would have more bias towards the prior. Okay? So if I increase this width, the force that is pulling me towards the prior will become greater. And I can do that by simply increasing the randomness in my stimulus, which will effectively make this F broader. And what you can see is that the bias is suppressed compared to the bias with more precise stimuli. Okay?
So at the 45 degree you have no bias essentially?
At 45 degrees you have no bias, yeah.
What are gabors?
Gabors are just this shape. It's like it's a sine wave multiplied by a Gaussian essentially. So it's just kind of like a small patch of grating, oriented grating. Okay, so now I want to return to this a little bit because there are some shortcomings, and that may answer part of your is relevant to your intuition, to the questions about your intuition, which is that we compared it with macaque data and it seemed to work, but if you compare it to cat data, you get a different result. Okay, so that's the result you get for cats. There are some issues of where exactly you measure cells and so on, but you get a much flatter tuning curve than you would expect, and you also get a flatter density of cells. Now, one thing that I didn't like about the approach before, I don't know about you, but was the fact of imposing a local constraint. Because the whole point of the calculation is to say, I have some global cost I want to minimize. Now I want to know how to allocate my resources locally at different positions in the stimulus space. But in addition to that, we had to assume some local constraint. And the reason we had to assume this local constraint is that otherwise we would have found that extremely narrow tuning curves will be, will be necessary. Now, remember, what we talked about is the cost of spiking in a neuron. But the spiking in a neuron depends on all the presynaptic neurons that are involved in making that neuron spike. So this is a very partial accounting of the cost. And how should we go about to think of the cost slightly more generally, where here is an experiment. They took mice and restricted their food, so they have low glucose. And what they saw is that indeed they use less energy. So this is the ATP consumption in the brain of a food-restricted mouse. This is the ATP consumption in a normally fed mouse. And what you find is that their brain do use less energy. Okay? How do they do that?
Proportionally less energy? If you cut in half, they cut in half, or still bigger?
I think they just did with one food restriction.
Okay.
So how does that happen is that when a neuron is excited, as I told you, in the synapse you will generate some incoming currents. And the currents that were generated in the food-restricted mouse was much smaller than the currents generated in the control mouse. And here you can see the size of the current as a function of the size of the stimulation by an electrode. Okay? So you would think that because the cell is less excited, it will fire less. Okay? And you save on the spikes that the cell fires less. And it's not at all what happens. If you compare the firing of the cells in the food-restricted mouse and control mouse, you find that they're about the same. So restricting the food did cause big changes in the neural signaling, but did not cause the cells to fire less. At least those cells under study.
So where is the energy saved?
Okay. So what happens is that, so there are various compensatory mechanisms which I don't know in detail, but what happens here is that the presynaptic cells become more variable. And as a result, even though the individual spikes will elicit a smaller response in the postsynaptic cells, because of this variability, the tuning curve will become broader. So what you can see is that these cells in the food-restricted mice would respond to many more orientation than in the control mouse. Okay. And as a result, the tuning curve in the food-restricted mouse will become broader. Okay. And this you can even see behaviorally. So they did a test. So you release a mouse, it has to swim. Mice really don't like to swim. There's a hidden platform where they can stand, and the hidden platform is in front of a target grating, not in front of the non-target grating. And so they have to learn which one is their target grating to get to the platform, not continue swimming. And when you compare the performance of mice, which are the control mice in black and the food-restricted one in red, their performance is similar when these two gratings differ by a big amount, but the food-restricted mice become worse when it's a fine discrimination task. And this suggests that this might be the cause of that. So they have less discrimination. Okay? So this was...
So that translates to less neurons participating in encoding the signal?
So, you know, I don't have a good... I would have to study it more in detail, but roughly speaking...
Because you say firing is about the same.
So
the only other explanation, there are less neurons, which also means that the discrimination is
no,
smaller.
so I think what it is that...
The amplitude was also the same, or maybe that changed.
Well, as I told you, this is the post-synaptic... No, the amplitude of spikes don't change. The
frequency doesn't change, so the only other thing is the number. I mean, according to your simplistic
it's more complicated than that. But the whole... No, but what I'm saying is that my simplistic model is not sufficient. That's the conclusion. What happens is that you have mechanisms that consume energy which will participate in the precision of the firing of the presynaptic cells, and maybe those are released. So
maybe they can't restore the synapse. You don't know. Maybe it's a long-term effect.
I mean, actually, they have studied it. It has to do with the pathway of processing leptin. So there is a specific pathway that they can address, which energy is saved into economizing on that pathway, which has to catalyze this molecule, and that will give rise to higher variability in the firing. So there is an answer to your question. I just don't know the details, but it's not of the simple kind you're looking for. So what this suggests is a modified version of the efficient coding prescription. I did this a couple of days ago, so you should check it and tell me if there's any algebra mistake. But so what I will modify now is I will add a cost of the width. Okay. So this experiment has taught me that if I give less glucose, I will have this bunch of mechanisms that will kick in and will give rise to a wider tuning curve. So I needed energy that I don't have because I don't have glucose to keep the sharpness of my tuning curve. So now what I'll do is I will add to this a term which will control for the cost of the width of the tuning curve. Okay? And I've put this cost of the number of neurons that I didn't like in brackets because I really don't need it anymore. So now all I will need...
Proportional to the inverse of it.
So I will not use even a function. Any function, there will be a large class of function that will give me the same result.
It's a
Decreasing function, sure. So this is now how my Lagrangian is written. This term is the same as what we had before. It's the total number of spikes. This term is the total number of neurons, but I don't need it. Here I will just have the sum over all my neurons of this cost, which is a decreasing function of W. Okay? The other change I've made is now I've taken seriously the fact that the variability in neurons is super Poisson. And so instead of having A here, I will have A to some power alpha. Okay? Which before we just simplified the variability as being Poisson. So when you do this, you get very similar result. you get that the density of neuron is proportional to the prior. But you get that the discrimination error is a non-trivial power of the prior. And if it's super Poisson, this power has the right sign. Okay?
Can I say, look, the body doesn't, as you say, doesn't solve every time the new optimization problem. So the mechanism is built there to handle the situation when there is a lot of glucose or there is little glucose. Okay? So there is a mechanism by which something stops working when there is less glucose, means the glucose is spread around, but if there is enough, it goes to all of them. If it's not enough, some of them don't get any glucose, right? I mean, what else can
I really wish biology were as simple as you think it is.
I know it's not, but you can't make it on the other hand as if every time a new problem is solved, because it's not. There is just one problem that is solved. That's what evolution has it, and you have to figure out how, in different situations, that same solution operates.
So it only partially for the following reason is that you have different mechanisms that kick in on very different time scales.
So you say that there is a change of phase between having a lot of glucose and having less glucose?
Absolutely.
It's a phase change. That's
Absolutely.
Okay.
And this kind of what you call phase changes happen on many different time scales. Happen on time scales of 30 milliseconds, happen on time scale of 300 milliseconds, on 3 seconds, 30 seconds, 3 hours, 3 days.
Okay. I mean, that's a different ballgame. Okay.
I will show, I don't know if I will, but I was planning to show some examples of that. Okay, now you get different solution for the width. You get a constant width and now an amplitude which behaves a bit weird.
So alpha is positive.
And smaller than 1, yeah.
So this exponent on the prior density cannot be -1, for example.
Cannot be minus 1.
It's something less negative than
Yeah.
exact.
Yeah, exactly. Okay, so now I have 15 or 18 minutes, so they're more than half left or something. So you have the choice going beyond simple models, where I will tell you about adaptation.
So you're not unhappy with that? You're not unhappy with the fact that it doesn't match the things you were showing us before?
You mean for which quantity?
They look like discrimination thresholds look like the inverse of the prior density?
No, you mean, discrimination threat? No, I'm not sure I believe really this, that it's so. Okay, so,
You want to vote on what to do? I will go to the last one.
the last one.
I don't know. You can't do all 3 of them.
No. Okay, I can tell you what is in which you can vote. This one, this one is a bit longer. I was going to, so before Sergio decides, this one is a bit longer. It has to do with just showing you how restricted these models are by showing you that things adapt all the time, change all the time, noise is correlated, and encoding is very complicated. This was to treat the question of non-ideal decoding, and this was a kind of preview for Mike's talk on Sunday, because it's using the same machinery. So maybe this is the shortest, so maybe I can do this, which is try to connect these issues with memory, which was the topic of the talk this morning. So Sergio decided. Oh, but let me just show you this, because it's amusing. So this is just to show you that things change very fast in time. So you look at this and fixate on this bar and move your eyes left to right for a moment, for like 30 seconds, and after just jump and fixate here. Okay? So what you should see is that these bars look like that, if you do it carefully. So there is some adaptation even on this short time scale of how your system works, and cells are adapting. And maybe one more thing. You can see here, this is the same as that, but it looks like that. And this, you were saying that if you have the messages coming from your menu should not depend on your menu. So this is an example where the message depends on the menu, And you also have attraction. So when, depending on how different these angles are, you can have either repulsion or attraction. So anyway, so let me go to the. That's a pity to skip this, but I will skip it. Okay, so let me briefly comment about how neural noise might be relevant to think about memory. And what we saw this in the morning lecture was that we imposed some precision cost, and that gave rise to noise in the way I update my memory, and that in turn gave rise to biases in forecasting. Here I want to say, what about how memory is implemented? And now I know that I have some neural noise. How should I think about that? Okay. So there are many ways that memory kind of is instantiated by the brain. There is memory on different time scales. There are something you remember for 5 seconds and then you forget. There are things like this lecture that you remember for the rest of your life. And these are, the substrate of these processes are different. One known process is that you have pieces of dendrite that grow and retract. You have chemical domain whose composition changes. You have many kind of synaptic changes. You have new genesis of neurons. So you have many different mechanisms that support memory. Here I'll focus on a mechanism that has been proposed and seems to be often realized in the case of short-term memory or working memory. And there it's assumed that, let's say I tell you a phone number, you remember it for 2 minutes or 10 seconds. You don't have the time to affect chemical or morphological changes in your synapse. And what is believed is there is some persistent neural activity that is generated in some neural system, and this activity is representing that information. Okay? So what you want is a system where you give some input and there's a record of that input that remains in the system in the form of neural activity. Okay? And so you can do, and now how would you measure the noise in that record? So this is one possible experiment. I show you a bar or 2 bars or 4 bars or 6 bars, but think of 1 bar for now. And then I mask it for a while. I did with a blank screen. And after a delay, I ask you to reproduce the orientation of the bar. Okay, so I'm staying in the same theme of the bars. Okay, so one system that has been proposed to support this kind of memory is what people call the ring models. You take a ring of neurons, and each neuron will correspond to one orientation of a bar. And if you build the synapses between the neurons properly, what you will obtain is that if I show you a bar, you will create a bump of activity locally in this ring, and this bump of activity will be self-sustained. Okay, so neurons will excite each other within the bump, will inhibit neurons everywhere else, and this bump of activity will survive after I've removed the input. Mind you, recently, such a ring of neurons was discovered in the fly, and it is a ring. So you actually have a ring, and that does suggest that you're saving on wiring costs. So it's, again, some kind of efficient coding prescription that would give you an actual morphological ring. Okay, now what happens is that these neurons are also noisy. They're not deterministic like all the neurons we've been talking about. And because there is some noise, and the noise is not symmetric, maybe this neuron will be more noisy than this neuron, it will push this bump around a bit. Okay? And so if you look at how the bump evolves in time, what you will find is that the bump is following some random walk, is diffusing over time along this ring in the absence of an input. Okay? And if you measure the variance of this diffusive motion, you find that it grows linearly in time. And so there is a diffusion constant, which is the ratio, which is the slope of this line. Now, as a quick remark, one can prove that this diffusion constant is related to the Fisher information. But this was proven algebraically for special cases, and to me it feels like a more general result that I think one should think about, because I don't know of a more general proof of a more conceptual proof of how diffusion is related to Fisher information. Okay. So now, one thing I forgot to say is that this diffusion will be inversely proportional to the number of neurons. Okay? So if I have a lot of neurons here, the noise will be averaged out. And so the fluctuations relatively to the bump will become smaller. If I have fewer neurons, each fluctuation will be more prone to move my bump. And it's very easy to see that this is linear, inversely linear in the number of neurons. Okay? So this slope should be inversely linear in the number of neurons. Okay?
Right.
Yes.
So the width of the bump is staying about the same.
Yes.
As it
Yes.
diffuses
Well,
over.
yes. The width of the bump is staying about the same. You have small changes, but it's fluctuating around the same. That's right. In fact, even if you move it with an input, the width, of course, changes a little bit, but it's kind of constrained. Yeah. Okay. So this experiment now was done, and the way at least this author interpreted it was that you're using a system with a large number of neurons, but if you have two items, you have access to the same number of neurons, so each item gets half the number of neurons. Okay? And so whatever the diffusion in memory should be for one neuron, if you can fit that, then you should automatically know the diffusions for the other cases. Okay? And so that's the experimental result that they find, which is that for one item you have very little forgetfulness, and for larger items you have bigger and bigger diffusions. Okay?
You are asked, in the fever, you are asked the orientation of
one of them. Exactly.
By
So
color, they tell you
exactly.
the green one
I
is
should have
something.
said that. Yeah, absolutely. Thank you. Exactly. Okay. So theory gives you something that is very similar. Now I'm cheating a little bit because this is not the one. It's one way you would fit it, which is not maybe the best way to fit it. But so the theory is roughly okay. But what you see is that there is some overestimation of the noise for small number of items. And one proposal that I don't know very well by these authors, but it would be worth studying, is that you have another layer of encoding before kind of putting it in a memory system. So you first have a compression, then you store it in memory, and they claim that this would explain this discrepancy.
In terms, going back to what you told us before, the total number of resources spent when I have one item or 6 items, it's the same or it grows linearly?
Same.
Same.
In their assumptions, yes.
No. There is no empirical measurement here. You only see what, it's a completely theoretical model,
That's right.
and you see what people do, that's all.
That's right.
Nobody measured that.
That's right.
Okay.
Well, the thing on the right is data, right?
That is data,
And the bars
and
on the left
these
are
are
all where it's
on
the
response.
same number of neurons.
same number of neurons. Now you could also assume
Data
that
on response, not on neurons.
yeah. No,
But on the left, there is a theoretical assumption.
but Sergio is saying you didn't record neurons to see how many are active. I'm saying this is all behavioral data.
Yeah,
That's
okay.
yeah. But it relies also on the assumption that you're using a ring model separately for each of the bars. You could have a torus model that would include two bars at the same time, but it would be more costly. So there is an argument for using one ring per bar, because the other schemes would be more costly.
We do have some control over our memory. That is, if something is very important, we can
Absolutely, yeah.
make an effort and succeed.
Yeah.
Right
Yeah.
down
the pattern.
Yeah.
That doesn't seem to be present in
That's right.
this description.
That's right. So there are various ways you could do that. In fact, one way is to couple this system to another system that will provide some form of information that is stored in another way. That's a very trivial way. But it seems to be that this is the way, for example, that you can refer to visual information when you're dealing with spatial memory. But another possibility is that you actually create a bigger well, potential well here, and you freeze your memory. So I will show you an example of that in a minute. And I will end with that. Okay, so now let's go to another experiment which is very similar. Now you're shown a bunch of squares of different colors at different locations. And after a delay, you're shown the location of the square and you're asked to choose what hue, what color it was. Okay? So here you're shown this location and you're trying to guess the pink. Okay? And what people, the behavioral result is as follows. So the black line are the experimentally used colors, and the shaded
Men or women who participated, it's very different population in recognizing colors. You know, men
Yes.
have only 8 colors. Women have 256.
Well, it looks like it was a man because it has 4. So you have this corrugation in the estimations of the subject. And so what has been proposed is that in fact this corrugation comes from the fact that you're not using a perfectly tuned ring model, but you're in fact using a model that has fixed attractors, fixed potential wells, and you're kind of diffusing in between these potential wells. And so the proposal here is that instead of encoding a continuous variable, you're in fact encoding a discrete variable. So let me go back to this image, where this bump could be at one of a few places. And the argument is that this will prevent you from diffusing infinitely far. All you will be able to do is to diffuse to the next potential well, okay, the next attractor, and that would indeed cause such a corrugation, okay? So if you take such a model, so now you take a model with 4 attractors and diffusion, so within the attractor you're diffusing, occasionally you're jumping from one attractor to another, this is the kind of outcome you would get. So you have 4 attractors, and this is the density of where your bump, the long time density of where your bump is sitting, okay? But note that this also has another perceptual prediction, because it says that if I start here, then I will have a high chance of ending here a moment later, okay? So if I now ask you to report this color, you will have more chance of reporting this color or this color than that color. And so this should give you a bias which is attractive when you're close enough to an attractor and then becomes attractive to the next attractor. And that is what is tested here. So these were empirical biases measured in people, the dark blue line, and the light blue line is the same biases for this very colorless man who can only distinguish 4 colors. Yeah. Okay, so let me finish and okay, so maybe just one comment. So here, just to contrast with what we did this morning we had a noise that was added upon either storing or retrieving a memory. Okay? In our model, storing and retrieving were indistinguishable, and we could assign the cost to one or the other. So we had to have some active mechanism that would cause this noise. This would happen at every trial in the model. So we could not really distinguish either in the model if this is an active mechanism that the noise depends on having retrieved the memory or stored the memory, or if the noise is just added on every trial. And what this kind of model suggests is that there's a natural diffusion of the memory whether you touch it or don't touch it, and when you touch it you can move it around, but you're adding this diffusion noise. Okay, so let me conclude just with 2 remarks. that maybe to contrast this picture, so again, as I was saying, one of my aim here was to try, and unfortunately I skipped the part where I was doing most of that, but to try to convey that it's extremely messy when you get to implementation. And I think the two big differences with the kind of more idealized models that we've seen, that I've talked about also this morning, is that there is really a large multiplicity of representations and they're related. So for example, if I ask which part of your brain is correlated to a stimulus, you will find many parts, many neurons all over your brain that carry information about a given stimulus. And so you have many replicas of some representation, and all these representations will be different and they'll be interacting with each other. Whereas in the more idealized models we tend to use, we think of a single representation of some quantity. And the other thing, which unfortunately I didn't have time to illustrate very much, are not only that these representations are noisy, but they're constantly changing over time. And in many cases, these changes have very different characters for a given representation, and also many different time scales over which different types of changes occur. So they're not fixed representations. Thank you.